Hello again. This is the second video in relation to signal transduction and the stress response. So once again, this one is going to relate to increasing the energy available within the cell. But we're looking at a different hormone. So we've got adrenaline here. Now, adrenaline is released from the adrenal glands. In this scenario, we have adrenaline binding to a receptor. Now that receptor will always be an extracellular receptor because adrenaline is a hydrophilic or polar molecule. It can't actually move through the membrane. So what you can see here is a G protein. So this is supposed to look like the letter G. A G protein coupled receptor that always spans the membrane. And what you'll see is adrenaline binding to that receptor. And we've got subunits on the inside of the cell, and the binding event induces the release or dissociation of one of those subunits within the cell. And in turn, what that does is it activates another protein called adenyl cyclase, which eventually initiates activation of this new molecule, cyclic AMP or CAMP, which is derived from, you'll see here, ATP. So that's what that represents here. ATP you'll be familiar with, but that's actually the synthesis of CAMP comes from ATP. You don't need to know the details of this process. What you need to do is get a general understanding of this process and use it for the activity that you're going to do in your booklet. So far adrenaline is bound to an extracellular receptor, and that binding event has caused a change in the G protein within the cell that's coupled to the receptor. That's activated adenyl cyclase, which has caused the production or synthesis of cyclic AMP from ATP. Now, here what we've got is another ATP coming in and another CAMP being produced as a result of activation of that adenyl cyclase. What that is indicating for you is the amplification of that signal. So a single adrenaline molecule binding to a single receptor can activate an adenyl cyclase, which in turn will initiate or activate a number of these second messengers. So the cyclic AMP here is an example of a second messenger. So we've got a couple of those, probably more than a couple within the cell being activated. Okay, the next step in this process is another molecule, so protein kinase A. Once again, not an important thing for you to remember, but it's another molecule in that transduction phase, so the signal cascade. And the result of activation of protein kinase A is that we are going to inhibit the production of glycogen from glucose, which would reduce the glucose available in the cell and store glucose in this larger glycogen molecule. So if you switch that off, you're going to keep glucose available within the cell, and also you're going to promote the breakdown of glycogen by these proteins that we've activated. And therefore, if you break down the glycogen into glucose, again, you're going to increase the amount of glucose available in the cell, and that facilitates the stress response. Okay, so you have a number of statements in your book out of order. I'd like you to look at that and try and just reorganize those to reflect the process that you've just seen. So the receptor, the ligand binding to the receptor, initiating a change in the G protein, one of the subunits activating another protein, adenyl cyclase, which initiates the signal cascade and activation of second messengers, which in this case have been various types of second messengers, and eventually activating a protein which causes a cellular response and eventually leading to an increase in glucose availability. Okay. You may watch that again if you need to.